Okay, so the last video for today is on the last part of the scientific method, which the next step is analyze the data. So we've already made an observation, conducted research, made a hypothesis, and tested the hypothesis. So now that we have tested the hypothesis, we need to analyze the results of that experiment. So you had to come up with some quantitative expression of your observations from the experiment. So here it just defines for you qualitative and quantitative. So you may have some qualitative data, but if you're reporting on that data, you need to quantify it in some other way. But as you analyze those observations, you could take those things like what color is it, how loud is it, what does it feel like, what does it taste like, take all those things into consideration. And when you make your graph, you need to have some kind of measurement expressed in numbers. The next step would be to draw a conclusion. So you're going to determine if your experiment gives a solution to the problem that you first identified in the beginning of the scientific method. So you're going to answer the conclude using two words: refuted, which means that your hypothesis was not correct, so you need to make a new hypothesis and repeat the experiment. If it's verified, you're going to repeat the experiment to be sure that you don't have an incorrect conclusion and that something did not go wrong in your experiment previously and you're drawing the right conclusion based on your information. So a conclusion is a judgment that is based on observation plus experimentation. So you can't just look at the sky and conclude that it's going to rain. You have to have an experiment and a series of observations to be able to conclude that accurately every time. The last step of the scientific method is to publish your results. You're going to have your peers review your final report and then publish them to a scientific journal or publication. So the more reputable your source is that you're published in, probably the more merit that your research is going to hold. So if you are a cell biologist and you publish in Cell, then it is probably going to be better than a cell biologist publishing in something like ecology. So you publish in your area of study, and the more you publish, the more you're funded as a scientist, and then you become famous, or as famous as scientists become, and that's how you make money based off of your publications. So now we're going to move into talking a little bit about the different types of variables. So we're going to skip some of this stuff here. So we have four variables: the independent variable, the dependent variable, the control, and then the control group, which isn't exactly a variable, but we're going to consider it in this group. So the independent variable is the variable that is changed or manipulated. So that comes in with our saying dry mix as we're graphing. The mix would be manipulated independent. Variable goes on the x-axis. So the independent is what you manipulate. You can only test one independent variable at a time. This is what is different in the experiment. The dependent variable changes as a result of the independent variable. It is what you measure. So if you're measuring how tall the grass grows as a result of fertilizer being applied, that's what you're measuring. That's your dependent variable. It depends on what type of fertilizer you used. So dependent responding variable goes on the y-axis. The control variable is the variable that is kept constant. It can be multiple different things. You'll also hear this called the constant variable. So if you're giving all the grass plots the same amount of water, if the size of the plot is the same, those are all of controls or constants across the experiment. And then your control group is the group you base your results off of. you do not apply the independent variable to the control group. So if we were testing fertilizer on the growth of grass, then the group that you would compare everything to would be a group that does not receive any fertilizer. So for more help with variables, scientific method, there are some videos on the Unit 1 page that may help you kind of remember. Don't forget that your note sheet being turned in is your homework and the grade that you will receive for this watching this video.